So, like as Alison said, my name is Rahul, and I lead the flagship genomics effort within CSIRO, which is Australia's National Science Agency. But increasingly, I also work to drive the development of our AI for Science platform, which is really focused on the accessibility. Once you have all sorts of fancy things, how do you actually make sure people get it? So before I start, I'd like to acknowledge the traditional owners of the lands that we meet on, and for me, that's the Wurundjeri people. Now, if you think about biosecurity, we have a few critical challenges within biosecurity. First thing, it's about, when you talk about R&D in biology and biosecurity and bioinnovation, it's more than just about humans. The human species is much more studied than the rest, but there are way more unknowns within the sphere of bioinnovation and biosecurity, and that changes with every single species that we work on. And right from the innovation down to the implementation and monitoring end, it is an extremely labor-intensive field, which makes it very tricky to move fast in the field of biosecurity because we're always playing catch up with the demands that are placed upon us, either as researchers, as practitioners, as communicators, or what have you. The other issue that we have, and we come across a lot in biosecurity, is the access to expertise across the globe. Usually, certain groups tend to be really, really good at certain things, but then they only have a certain amount of manpower or a certain amount of labor resources or what have you. And so it becomes really hard to access this kind of expertise globally, which also slows down the biosecurity responses that we can have and/or any biosecurity innovation as well. And lastly, life in biology is multimodal, which sort of links to the first, sort of the second point that I made earlier. And what I mean by that is life, when it comes to bioinnovation, biosecurity on the biological front is about DNA, RNA, proteins, metabolites, and so on and so forth. But it is also about looking at observations, text, images. It's a very complex system. And so we need to take all of that multimodality into consideration, which makes this field exciting, but also, again, it used to be slow to accelerate in biosecurity. This is sort of the time when large language models and AI models sort of started to come into play. And so just to sort of sketch out what I'll be taking everyone through today is I'll set the scene for AI. And by AI, I mean more around the generative AI. I think the speakers on the call are much better at the machine learning AI than I am, and I'll let them introduce that. But also how we apply it to real world situations to make sure that we can actually accelerate R&D, accelerate outcomes, implementation and monitoring, if you will. So I think everyone knows about ChatGPT. It's probably one of the most used words these days. That is about 2023, 2022. What we have seen, that was the first sort of paradigm shift, if you will, in human use of information. But it wasn't very useful when it came to actual real world applications beyond writing. What has happened, however, since 2024 is what I sort of consider the second paradigm shift. And we're currently going through that. And that's really the beginning of what we call agents. Agents is essentially, if you think about large language models or ChatGPT as just interacting with you in one stream, agents can actually have multiple ChatGPT conversations with each other and use tools and actually solve problems. And that's what makes it a lot more reliable and a lot more steerable. And that's important in biosecurity because we want to make sure that whatever we use our technologies for, the end outcome is reliable, repeatable, steerable, which means it does what the intervention is meant to do and not just random generation. And so what we're really trying to do with science, with implementation of agents in science, is we're trying to make these autonomous sort of or semi-autonomous sort of agentic systems more accessible to people. If you think about it in terms of Lego blocks, each of these systems is a Lego block, and you can build a lot of things by just putting Lego blocks together. And what it really looks like in practical terms is you have the AI glue, but the AI is essentially just a way to translate information from models to tools to data input and to interface and to eventually to intervention. And that's really where the generative AI field or the artificial intelligence field is really pushing nowadays, is implementing all of these wonderful innovations that we've done into tools or models so that we can have a better sort of handle on what we do with our data and what sort of outputs we generate. Now, again, taking a step back, the scientific research process, when it comes to any field really, uh, consists of 5 steps the way I see it. The first is to ideate and come up with the hypotheses. The second is to scout, which is find out a bit more about the topic, find out a bit more about who does what in this area, those sorts of things. Usually that leads to project proposals, and then we generate data within that, and we analyze it, and then we share it, and we implement it. That's the usual scientific process. What we're trying to do is within our teams and broadly in the community is we're trying to turbocharge and scale this scientific method significantly by automating a lot of these things. And what you can do really is you can automate a large part of distillation of your thoughts, and that's, we generally pretty good at putting our thoughts down on paper or in conversation. How do we actually bring it all together? How do we bring all our hypotheses together into one ecosystem so that you don't, if you will, it acts like your second brain, right? What we want to do is we want to develop sidekicks using these novel AI and the new AI technologies so that we can remember better, we can study better, and we can deliver science and develop science a lot better. And that's both, that goes all across health and life sciences, genomics, biology, biosecurity, what have you. But again, coming back to the tangible question of the day, right? What does this have to do with biosecurity? We can develop agents, we can look at agentic systems, artificial intelligence. How do we actually implement this in biosecurity, and how do we actually scale? What does it actually mean? And so, some of you who might be aware of this area, I think there's lots of efforts around, such as Google and Sakana and so on and so forth, where they've been trying to develop sort of the co-scientist systems, co-scientists that are focused more explicitly in the human area, but they aim to accelerate things like drug development, machine learning, and so on and so forth. And that's sort of really where I see the future of biosecurity being going, because it takes care of a few very key challenges that we have faced so far, such as manpower, dissemination of information, sharing of insights. And so if you look at the standard sort of use case, in this case would be, let's say, developing a gene drive or developing an RNAi intervention or developing a biocontrol method. It usually consists of a few different steps, such as you'll assemble the genome, you'll discover all the genes, you'll discover targets for RNAi or gene drives or what have you. But usually the critical point is you can find lots of targets. There's lots of softwares to do this. But what does it actually mean? What are the functions? What is the actual intervention? What is the impact of the intervention? What does that translate to in real terms? Will this biocontrol work? How will it spread? What will it do? And finally, also reviewing literature and disseminating information. Those are the kinds of things that become quite important. And we use these routinely in our projects within our teams, where we've got machine learning and AI-driven models or systems for all of these. But I'll primarily go through the bottom three because I think these three are the most important in terms of actually unlocking what biosecurity needs, which is speed, accuracy, reliability, steerability. So the first one that I'll go through is gene function prediction. And our gene function prediction for us is quite important because our team, for example, develops a lot of genetic biocontrol strategies. We do a lot of surveillance across populations for invasive species to study how they move and what the impact of their movement is, what the adaptation is, or what adaptation is actually driving invasive species to move across the line, or even identifying what genes and what genetic properties actually contribute to a certain species existence, such as, if you will, biodiversity assessments and things like that. And so we use it very extensively in developing genetic biocontrol strategies. It is very critical for us because most species that we work with, we know nothing about. Most invasive species have very little knowledge in terms of functional evaluation of the genes, and even any experimentation work. In most cases, there's actually no data, and we're starting from scratch. And the reason why this is important is, if you think about many of the people that have worked in this area, if we go down, let's say, a certain species, and we're trying to identify function for a certain phenotype or what have you, or the genetic basis of a certain phenotype, it is a very labor-intensive task. And more often than not, the labor expenditure, the time spent, is in reading papers, gathering databases, things like that, which is incredibly important when it comes to increasing the precision, but it slows down. And what we wanted to do was essentially break this cycle. What takes, in a classical PhD, something like this takes weeks, sometimes months, sometimes years, depending on complex things. We wanted to bring it down to minutes. And that's what one of the work, that's what one of our postdocs, Maisie, has done, is she's developed a system called Gene Whisperer. And what Gene Whisperer does is it's a complicated multi-agentic workflow with lots of different models incorporated into it, lots of different databases linked into it. And so when you give it a simple thing such as a sequence of a protein or sequence ID based on NCBI genes, it will actually go and read papers for you. It will gather, it will run blast searches and protein structure searches and things like that, and find you all the information that is actually required to ascertain what this unknown protein might be. And it might be. We're not making, we're trying to make sure that we don't give very prescriptive answers, because AI is still sort of catching up with the real-time requirements for accuracy. But what it does is it reduces a week-long task into a 30-second query, and then you walk away. And I'll show you how we do this in a second as well. And this allows us to evaluate novel genes very quickly. And so for biosecurity and bioinnovation, bioprotection, this is really critical, because we can then do 30,000 genes for a new species in a day. And then all we're doing is actually sifting through information and categorizing, which is actually the rewarding part of the journey. The second aspect that I'd love to take everyone through is, we can find the genes, and let's work through a hypothetical situation. We've generated all the genomic data sets, so we have access to them. We want to develop things like RNAi or gene drives or what have you. But with any single biocontrol technology, the most important thing is making sure that the target that you aim your intervention at has high efficacy, it is environmentally safe, it has limited off-target effects, and all of these things contribute to either knowing whether this will have a clear regulatory or decision-making framework or not. And the way we like to do it is try and bring a bit more of a genome-guided precision into this by studying populations and variation across populations. Now, if you think about all of these things at once, it's actually a really arduous task. A single gene target for RNAi or gene drives should take weeks, months to gather all this information, run all the models, develop all the models. And that's another opportunity that we saw, where we could actually use artificial intelligence, use large language models and classical machine learning to both run the models, evaluate targets, and deliver information to users in a way the user needs. And the users can be a wide variety of users. They can be scientists that are trying to develop this. And so for scientists, what they need information on is, what is the target? What is the function? How will it spread? How does it interact with this ecosystem? Things like that. The regulators have a completely different question. They want to ask things like, is this going to disrupt populations or native populations if it escapes? Is this going to create any other sort of unforeseen ecological and genetic disturbances in the populations? Things like that. And so we built this platform, and I think Heng spoke about this work that we did in the last session. And so please go and see the earlier talk on genomics as well, because he goes into a bit more detail with this. But at the face of it, what we did is a few years ago, we developed a platform that sort of hyperscales the modeling aspect of it. And so what we could do is, the models that usually take days to run, we managed to get them running in seconds. This is work by one of our PhD students and our team. And we could generate a lot of modeling results. But when we went and spoke to the regulators said, look, you can generate all the modeling results you want, and we really want to see modeling for every single target, but we don't have the time to sift through all of your models. So that brought us another opportunity, saying, okay, cool. How can we actually train our systems, train our agents to actually start helping regulators assess this information, evaluate this information, and make informed decisions that they need to make, so that we can then proceed with the innovation. And so we developed an addendum to the GUARD system. So we call this whole system GUARD, which is really aimed at, it was started off with gene drive, but it works for any genetic biocontrol mechanism that you can think of. But we turned it into a much more simpler system, where you can now say, run a gene drive simulation for, this particular thing in the rabbit genome and the target. And it will also obviously run all of the simulations for the different types of gene drives that we study in our team. But the other thing that it does is, the model is explicitly trained on all the input parameters, all the modeling parameters that are actually being incorporated in this model, but also all of the population demographics, history, and so on and so forth. And so what that allows us to deliver to the regulators and to the decision makers in the government, as well as R&D folks, is the actual insights. It's not about giving people massive images, reports, and things like that, because nobody has the time to read a 30-page report on a single target. But what they have, what people are really good at, is coming up with questions. So we wanted to create a system where you can ask the right questions and get the information from the models. And so what in this example, for instance, what it was doing is it reads every single generation that the model runs through, and it is continuously creating that sort of time lapse of how the gene drive will spread across a population, and then giving the user a snapshot. So this one is designed primarily more for government stakeholders and what have you. But this again becomes a massive unblock, right? It's not just an unblock for the scientist, because the scientist can actually use such a tool and come up with come up with insights that will incredibly help them uh, develop the solution further. But by sort of seeing what the regulators might be looking for, but it also helps the regulators because now it becomes a live conversation. We could almost have such a system deployed while the projects are running, with regulators, with scientists saying, cool, we did this, look at this. Do you think this is safe? If there's any concerns that come up, it saves time, it saves effort, it saves money. But more than anything else, it makes sure that the intervention that you're creating is targeted and safe. And so while we would, again, we got to this point and we said, okay, cool, the data generation was working well, modeling's working well, scaling is working well, inference is working well, but we still had not achieved that level of systematic automation and enabling outside of our team, because all of these insights again stay within our team. And that comes back to the last challenge that I mentioned, right? Which is within biosecurity, one of our biggest challenges is access to expertise. There's very limited expertise globally. We want to make sure that things are accessible to everyone. And there's a paper for the study, for example, done in Europe, which looked at how many domain researchers are actually using artificial intelligence or know about artificial intelligence. And I think the number was 18%, which is quite low. And so we wanted to come up with a solution for this. And sort of one of the solutions that we came up with was actually turning these agents into teams of agents and getting them to do stuff for you um, in a human guided manner, so that we can automate the link between the different tasks. So you can have you want to do genome assembly, you want to do genome annotation, function deciphering, finding targets, modeling, inference from the modeling, creating sort of reporting for all of these things. We wanted to make sure that the users can and scientists can actually access all of this in one place, and create a way so that people and scientists can actually develop different kinds of solutions and actually throw it across the line and make it accessible to everyone, because it seals and it creates a snapshot of human intelligence in space and time, which is quite important. And the benefit of these teams of agents is once you create one, you can make them talk to each other, and that's quite powerful. And so if you look at the scientific process, right, and or any project process, it consists of a lot of different things, such as finding experts, collaborators, patent reviews, brainstorming, proposals, experimentation, things like that. Um, that can be organized into two sections, essentially. One is evaluating the problem, and the second is evaluating, and actually, the second is actually solving the problem. And what these multi-agent teams look like, if I was to give you an example, is something like that one of the topic overview capabilities we have within our platform, which consists of both of three steps, which is a literature review agent, an analysis agent, and a synthesis agent. And then they can go back and forth until they satisfy the query, which is, it can be something as simple as lignin degradation in bacteria, or develop a new biocontrol mechanism. Let's find some gene targets for biocontrol in X, Y, or Z. And what we want, and we wanted to make sure that this was incredibly accessible to users. And what we realized is that this led in many ways to a platform that can allow users to put these teams of agents to work, ensure that every single step is highly triaged, so the user has complete control over what the task is. Each of these tasks has a complicated set of tools and softwares and modeling workflows and things that are running underneath it. But it ensures that the machine doesn't just wander off, and that humans are always in control. And in spaces such as bioinnovation and biosecurity, that's very important, because what we wanted to enable was that you can come up with a query, come up with a curiosity, lodge it into the platform as a query. Let the agents do their job and gather information for you so that you can spend 20% of your time doing 80% of the mundane tasks that you have, either as scientists, as leaders across the industry, as implementers, and use that 80% time to actually do what matters. Focus on the outcomes, focus on the implementation, focus on making sure that the science is actually done properly. And so that's sort of our approach, the approach that we've taken. And I think that's the promise that artificial intelligence brings to biosecurity, is both allowing us to increase to a drastic degree the confidence and the accuracy in individual steps, such as gene function prediction and modeling inference and so on and so forth, but also bringing it all together into an ecosystem in a manner in which we create a snapshot of people's expertise and uh, automate that unit component so that users can use their creativity, scientists across the world can use their creativity to bring these Lego blocks together and turn it into a solution, and every solution is bespoke. So I'll leave it at that. Thank you so much for your time, and thanks a lot, Alison, for inviting me as well.